Cells and microscopy. Organisms can be eukaryotes or prokaryotes. Eukaryotes, e.g. all animals and plants, are made from complex cells called eukaryotic cells. Prokaryotes, e.g. bacteria, are smaller and simpler cells called prokaryotic cells. Both types of cells contain subcellular structures, parts of cells that each have a specific function. In an animal cell, there is a nucleus, which contains the DNA, genetic material, in the form of chromosomes that controls the cell's activities. It also contains a cytoplasm, which is a gel-like substance where most of the chemical reactions happen. And mitochondria, these are the sites of cellular respiration and contain the enzymes needed for the reactions involved. A cell membrane, and that holds the cell together and controls what goes in and out by providing a selective barrier. They also contain receptor molecules that are used for cell communication. And in a plant cell, it has everything that an animal contains, but with a few extras. A rigid cell wall. This is made of cellulose and gives support for the cell. Chloroplasts, where photosynthesis occurs, they contain a green substance called chlorophyll. And within prokaryotic cells, there is a chromosomal DNA in this bacterial cell. A chromosomal DNA is one long circular chromosome. It controls the cell's activities and replication. It floats free in the cytoplasm. Plasmids. They are small loops of extra DNA that aren't part of the chromosome. Plasmids contain genes for things like drug resistance and can be passed between bacteria. Cell membrane controls what goes in and out. The cell is also supported by a cell wall. Cells are studied using microscopes. Microscopes use lenses to magnify images and make them look bigger. They also increase the resolution of an image. This means they increase the detail you can see. Resolution is how well a microscope distinguishes between 2 points that are close together. Light microscopes were invented in the 1590s. They let us see things like nuclei and chloroplasts. Electron microscopes were invented in the 1930s. They let us see much smaller things in more detail, like the internal structure of mitochondria. This has allowed us to have a much greater understanding of subcellular structures. Only electron microscopes will let us see things as tiny as plasmids or viruses. Transmission electron microscopes, TEMs, have a higher magnification and resolution than light microscopes, but they are not portable. They are expensive, and it's a complicated process to prepare specimens for use, which means they can't be used to look at living tissue, unlike light microscopes.